Hi, my name is Kourosh, Principal Bioinformatician, and welcome to Genonaut. Genonaut is an interactive platform for exploring single-cell RNA sequencing data. It's designed so that anyone, from a wet lab biologist to an experienced computational researcher, can dive into complex datasets without needing to code. This is the Gene Expression Explorer, the starting point for navigating your single-cell datasets. On the left, you'll see the main control panel from where you can choose how you want to explore. We can visualize the expression of individual genes, pre-loaded biological cell states, or custom gene signatures, either via UMAP, Violin, or box plots. On the right, Genonaut stores an image library of your generated UMAPs and plots, so you can quickly toggle between past results and compare them without needing to rerun the analysis. For example, upon launching this breast cancer dataset, I can see the entire UMAP sample of all 72,000 cells colored by cell type. From here, we can dive into our analysis, all without needing to write a single line of code. In the Gene Visualization tab, I can type in the name of any gene of interest and instantly check its expression across my different cell types, either through a UMAP or a Violin plot. This is the fastest way to see whether your biological target is enriched in your cell type or subpopulation of interest. Your Image tab can store up to 4 main images. Each image has the option to be enlarged, exported to your local machine, or removed from the main view. Removing an image will free up a slot to load a generated figure from your image library. You can drag and reconfigure the order to your liking. The box visualization is designed for more quantitative comparisons. It generates box plots with statistical readouts so I can directly compare gene or signature expression between groups. These comparisons can also be grouped by patient metadata that was provided for your dataset. Signature visualization lets me test entire gene sets at once. I can bring in my own custom signatures, and Genonaut will calculate a composite score for each cell. This will show me in what cells those genes are enriched in, which makes it easy to tweak and identify the most relevant subpopulations of interest for downstream analysis. The Cell Type Distribution tab gives me a snapshot of how many cells are in my original dataset or active subset. It's a quick way to check the balance of populations and confirm I've got enough cells for downstream analysis. Finally, if you want to explore clusters more deeply, you can adjust the lead-in resolution here. This lets you split or merge populations without needing to reprocess the dataset on your own computer. Each lead-in iteration will give you a new UMAP that you can annotate from analysis via the entire Exploration tab. The Analysis section is where Genonaut lets you create and manage cell subsets. You can filter cells by individual genes, signatures, cell types, metadata, or even by the new lead-in clusters you've annotated upstream. The benefit of this tab is that you can combine multiple subset filters at once. For example, I could create a subset of the top and bottom 10% of periostin-expressing epithelial cells, give that group a name, and instantly use it for downstream visualizations. Subsets can be saved, updated, or deleted at any time. At the top of the Exploration tab, you can always see what subset is currently active. You will always have the option to toggle between any subset you created during your session, or revert back and start over with the original dataset. The cell type distribution will always be updated to reflect what subset you are currently selected. Once you've defined your cell subsets, Genonaut makes it simple to run analyses directly in the browser. For example, we could perform a marker analysis, which identifies the top genes enriched in your grouping of interest. You can select the number of genes you wish plotted, as well as adjusting the p-value and fold change cutoffs. You also have the ability to run a secondary analysis. We can pull the genes from our periostin high epithelial cells and run a gene set enrichment analysis using the pathway libraries available. All of the plots and CSVs that have been generated in the Analysis Results section can be downloaded to your local machine by clicking on the respective Export button. After running your analyses, you will always have the option to go back to the Exploration tab and generate new UMAPs and plots using your cell subset. Our AI Gene Insights can be called upon at any time to learn more about genes you come across during your analysis session. A general summary regarding the gene and its function will be provided, as well as links to informative resources. And there you have it. An analysis that would have taken an informatician a couple hours to do, we just completed in 5 minutes. Our goal is to help democratize single-cell sequencing analysis for you and your research group by making it as straightforward as possible. Thanks again, and we hope to see you using Genonaut in the future.